Thanks very much for having me today. I'm looking forward to giving this talk to you and letting you know some work that I think we're doing that's really putting Utah on the map. I do need to let you know that I'm going to be talking about suicide risk today. This is a really difficult topic, and I want to always put this slide up here if either you or anyone you know is experiencing any distress in this area. We have some great resources. There's a new national crisis line. There are also local resources and services, and I'd encourage you or your loved ones to reach out. So, not a big surprise. Suicide is a huge and growing public health crisis right now. Suicide risk is especially problematic in the state of Utah, where we tend to be in the top 10 states in the country for suicide death. And so this is a crisis that we really need to address, and we have some resources right now in Utah that allow us to start doing this and to really get at this very urgent problem. So in Utah right now, we're really studying risk factors that lead to suicide death. Well, why would we study this? Right now, there are studies worldwide that are looking at risk factors for who attempts suicide. And a suicide attempt is currently the best predictor of who goes on to die by suicide. However, fewer than 10% of people who make an attempt will go on to die by suicide, and more than 50% of people who die by suicide don't have any evidence of a prior attempt at all. This is from a big study from the CDC, and actually our data in the University of Utah show us this as well. So really, our ability to predict who dies by suicide is not very good. And we also know that genetic risk is very important for leading to suicide and suicide death. And so in Utah, we have some resources that specifically allow us to address that very important aspect of risk. So right now, as you just heard in the introduction to this talk, we have some resources that are really unprecedented. Through a collaboration that was started by a colleague of mine a couple of decades ago, we have over 8,500 DNA samples from individuals in the state of Utah who died by suicide. This is through a terrific partnership with our Utah State Office of the Medical Examiner. And worldwide, there is no other research center that has this kind of biological resource of individuals who've died by suicide. We also have another really unique resource. So we can link that interesting genetic resource to something called the Utah Population Database, which is a statewide database with over 11 million records from individuals. It includes longitudinal health records data back to about the year 2000. It includes demographics. Also, interestingly, it includes genealogical records from individuals with ancestors back to about the 1800s, sometimes even 1700s. So we can construct very high-risk genealogy records of various health conditions, including suicidality. Really importantly also, we have people. So we have very collaborative groups of individuals who want to study this really important risk factor. These are people at the University of Utah, but there are also people at Intermountain Health, there are people at the health department, and there are people within our community that want to help us. So since our main research focus right now with our study is genetics, I want to give you a little bit of an update on what we've been finding. That's, of course, only one aspect of risk, and we know that the genetic risk is likely to be incredibly complex. We have these couple of ways of really looking at risk. First one is these big genealogical records that we can tap into. And what you're looking at here is an example of a family. So this is a diagram of a family that goes back to the 1800s, that the folks up there at the top who are the founders of this big family tree. And you can see at the bottom there, so anybody in this family who's shaded in black is a person who's died by suicide. The people that are circled in red are people where we actually have DNA samples. So by looking at a big extended family tree like this, we can try to determine areas of the genome that these individuals share. And that may lead us to genes that have an increase in their risk for this outcome. Also, because we just have a very big sample, we can look at other kinds of study designs, such as association studies, where we just look at the DNA across the genome from all these different individuals where we have their information and compare to a matched control sample. Okay, so what have we found so far? Well, it's complex. Maybe not surprisingly, we have some signal that suggests that there are some genes that are involved in risk that have to do with neuronal development and neuron function. We also have some gene findings that look like they are showing us changes in genes that are also associated with co-occurring psychiatric risks, depression, anxiety, PTSD, and developmental delays, and neurodegeneration as well. But we also are getting some other discoveries that are interesting to us. So there are some gene changes that look like they're involved in risk of suicidality that have to do with inflammation, that have to do with the immune system, and that have to do with energies to cells. And we also have some gene changes that look like they have to do with the way genes are regulated, and in particular in response to stress, which might, again, not be too surprising. So we have these studies, but we want to make sure that we really are interacting well with our clinical colleagues so that our findings are really clinically relevant and that we understand how to interpret them in the context of care delivery. So right now, because we have such a large population-based sample, any particular clinical subset or even demographic subset that a researcher might be interested in, it's in our sample. So right now we have groups of individuals who are studying suicide risk in youth and youth specifically exposed to trauma, in individuals with various conditions such as bipolar disorder and postpartum depression and other things like autism. We are trying to study demographic groups. We have this ongoing study looking at risk in transgender individuals, for example. So genes, of course, are only one part of the picture. We also think it's very important to study genes in the context of other stressors that are social stressors, environmental stressors, and this being across the lifespan. So any individual genetic change needs to be studied in the context of whether it is enhanced or repressed by other kinds of interacting factors. And I'm not going to go into this in too much detail, but just to say that our group incorporates individuals who are experts in epidemiology, in demography, and in thinking about social determinants of health. And finally, we talked a little bit about this in the ethics panel across the hall. We really are focused on the ethics of what we're doing. So in a very proactive way, we want to understand the oncoming potential for a genetic test for suicidality, genetic tests probably, plural, for suicidality. How will those be received by our stakeholders, by the people who are survivors, by family members? So we have engaged in some ethics studies to understand all these ethical implications. So where are we going from here? We have a lot of resources. We're really on the map in Utah for trying to address this very critical issue. And we're hoping at some point to work towards a really integrated sort of programmatic institute for looking at suicide risk and really getting at this problem in a good way. Something akin to the cancer moonshot that was done in the past decades. And of course, we hope to save lives. So I'm an optimist, and I hope that at some point our resources will get to some solutions here, but even if we save one life, it will have been worth it. Here's some of the individuals who have helped support this work, and many collaborators. I'm hoping at some point that the font is so small on this slide that we can't even read it, hoping to bring all these people in and help us with this problem. Thanks very much.